what I wanted to do was to give a status report of what's been going on with my researches as well as pointers for future researchers. I just turned 70 in November. I'm having some issues. I got knees replaced, and I'm still learning how to run my or use my legs. And so going to places like Mist is difficult. And there, every time I'm done with this locality, I think I'm done. But then something new comes up and stuff. So anyway, well, let's get right on. Of course, everybody in here should know where this is. It's up on Highway 47 along the Nehalem River on that somewhere on that bluff. Um, the Can you guys see my pointer?
Yes. Yes.
Yeah.
Yes.
There's a big long landslide chute that you can see from the highway that ends up down here. That's about where most of the fossils are found in this vicinity. And the biggest puzzle that I've had for since 1970 when I first went here is why do we only find these fossils here instead of all over this bluff? Of course, this is the thing that everybody wants to find there. This is the holotype of Isocrinus oregonensis. This is actually a mold of a specimen, um, and I'll talk more about that. This is at the U.S. National Museum of Natural History, and I believe it's on display. This is out of Warren Volk's 1951, the paper that Larry brought in. Mist is up here. There are other places in the Keasey Formation where crinoids have been found in the past. The only one I've found is actually the Haydite quarry when we used to collect, which is down here near the railroad trestle. You're no longer allowed to collect there, especially into the quarry. The railroad cut that is just southeast of the quarry. It's possible to find fossils there, but it's well overgrown. This locality is the storage locker just outside of Vernonia. And I've actually found a few articulated crinoids and a lot of crinoid parts and also crinoids associated with sponge fossils. Of course, here's our locality up here. This, of course, is the legendary 1951 paper. You can actually download this from the USGS just by simply searching for the USGS professional paper 233E and just follow the links and you can download it as a PDF. I started working on this locality in 1970 in my freshman year in high school. I was a student at the Student Research Center at the Oregon Museum of Science and Industry, and this is one of the two papers that I did there. And along with it came a lot of preparing a whole lot of fossils and identifying a lot of stuff. And then in 2005, um, um, we published this. It took a while for the science to catch up. We didn't even know about methane seeps, and although we saw various patterns of stuff, and it finally the science caught up. Another thing was observations of modern crinoids and the fact that they don't like to stand on something soft; they like to stand on something hard. Um, and so I thought, wow, there might be limestones there at the locality, which is what these things were standing on, and the limestone might be caused by methane. And this is sitting on top of one of the only commercially producing methane fields. So we went and we tested the the isotopes of carbon and oxygen, the stable isotopes, and there was some signs of methane seep activity, and but not something like there are two other methane seeps in the Keasey Formation, and these were not as significant change from just normal calcite. So it means that there was more work to be done. This was by just a preliminary report, hampered by the fact that I'm not a professional paleontologist, and the professionals that I'm working with are very, very busy with many, many other things. So it's just been just kind of my avocational passion project. Additional work has been done trying to pin down the age of this. Is it Eocene or Oligocene? We don't know. The paleontology suggests that it's Eocene, but the magnetostratigraphy actually has it in this magnetic reverse zone. If you don't know what this is, this is our north and south poles essentially switching back and forth through geological time. And the boundary of where the Eocene and Oligocene is currently set at 34 or 33.9 million years. But it may be that, there's places where the Eocene essentially persisted into this boundary and stuff. So the paleontology suggests warmer water. And it was soon after Mist that these stalked crinoids disappeared uh, from these seas entirely. They're found in the warmer waters of the Western Pacific and the Bahamas and such, but the isocrinids aren't found at all on this coastline. More work is being done. This was a 2017 master thesis done by a friend of mine, Mahiro Matsumoto, who's one of Tatsuo Oji's. Who Dr. Oji is one of the world's great crinoid researchers. She did some experimental work with modern crinoids and compared them to how they preserve with fossil crinoids. Is everybody with me still?
Yes. Yes.
Good.
Yes.
Okay. And then Carol Hickman, this is the title page of one of the four papers on the bivalves of the Keasey Formation that she's working on. She still has one more paper to do. She's 82 and would like to have the time and energy to get it and about, maybe 5 or 10 other papers done. Um, so this particular fossil she named after me, Saxicavella burnsii. It's one of only a few specimens that we found of this. So here's one of the other holotypes or paratypes of the crinoids at Mist. I'll talk about the crinoids first. This one is one of the ones, I think this is one of the ones at the Smithsonian. They had access to material that essentially was unweathered original calcite. You're probably more familiar with these beautiful specimens such as this, where you get the blue calcite on top of the tuffaceous matrix. These are very lovely. They're gorgeous specimens. They require either a ton of handwork or an air scribe to use. This one was just after I used some talc to highlight it, which actually the talc increases the contrast a little too much, and I didn't fully blow this stuff off. This was one of the specimens that I prepped out in the early 1970s using an SS White air scribe, which wasn't properly vented. I gave myself kind of bronchitis throughout my entire high school years. This is what you more typically collect. This material is almost totally unaltered. Um, it's the original uh, specimen, whereas this material has some sort of chemical alteration that has happened after deposition, possibly from the seep activity. The geochemistry of that is yet to be studied. That's yet another thing that somebody could do. Why do these things go from a lighter colored specimen to that? We always kind of thought that these were weathered specimens, and we were looking at things in reverse. This is one of the specimens that I collected just before we had a big slide there in 1995. You recall that big landslide chute that comes from the top. The summer before, we had the neighbor or the owner of the land on the top using a large tractor trying to, or bulldozer trying to remove stumps, and he almost backed over the top of the cliff. Mr. Epling, who owns the land across the stream, saw him up there and went up there, and they actually had to winch the tractor back. But then that winter we had all sorts of rain and snow, and it melted quickly, and there was a loud sound, and some 30 yards of material fell down. Well, that is, or not 30, about 10 yards of material out from the bluff. I'll talk about the conditions in a bit. Crinoids are still around. These are modern crinoids. These are some that are probably from the Bahamas. Notice the light color. That's the original color of these things. Um, here's one called Neocrinus. This is found in the Western Pacific. This is off of our shores. This is Florometra ceratissima. These are a stalkless crinoid that moved in. These they find stalkless crinoids all the way up into the Arctic, and these are found in water. You have to get down to about maybe 75 to 100 meters before you start seeing them. Although you farther north you go, the shallower they get. And like up in southern Alaska, there's been reports that you can snorkel down to some of these things. We've had crinoids for a long time. This is a slab of crinoid columnals from the Chilliwack group, which is, I think, Devonian or Mississippian age from up in the northern Cascades. This particular locality is the riprap at a parking lot at Safeway in Bellingham. And then just northeast of Seattle in Arlington, the person who did my gutters, he brought this specimen over. This is a very nice boulder that was found at a Army campground, Army-owned campground in forget the name of the creek, Salt Creek or Robinson Creek or something. But yeah, he just found this laying in the bottom of the stream when the stream was all dried out. It's a very lovely piece, but look at the size of these columnals. They're almost as big around as my friend's thumb. So
We
this,
have found some of those in the grindstone terrain out in Central Oregon as well.
yes, I've heard about those out there. Also, if you go up to Friday Harbor, and look at the former lime kiln that was there, you'll see them. Here's another specimen of crinoids, Neocrinus, that was from the Eocene west of Port Angeles, found by Jim Geddert. This is an interesting specimen that can show you the amount of time that it's required uh, between the discovery of the specimen, which this one was in the early to mid 1990s. Just last year, the paper came out. Going from finding and discovering and preparing a specimen to the actual discover or to the description can take forever, which is one of the reasons why I am not a taxonomist. One of Christina Franz and Bengtson, she was sent this specimen and she just sat on it and then did some preliminary work, and then eventually it was sent back to the LA County Museum and then forwarded to Tots. So I don't think Christina is even around anymore. But these are more modern crinoids. One of the things that I'm curious about is how these things lived and how, what were the conditions, and, what kinds of signals do we see, various arm postures showing various things. These are some stocked crinoids off of Florida that were imaged by Chuck Messing and others, taken with the Johnson Sea Link submersible. If you go onto YouTube and look for crinoid running, you'll see one of these things running on the ground, dragging its stalk, which doesn't have any muscles, with its arms, which has like 5 sets. Here's more crinoids. These things basically, their heads or their mouths and stuff are here, and these bring in various nutrients and stuff with the various pinnae and stuff. I won't go too much into the biology of these things, but they're beautiful animals in the deep sea. They're not something that you can see from a dock. You have to go down pretty deep to find these things. The Okeanos Explorer and some of the other submersibles frequently encounter various crinoids. Here it's back to the Mist. This was one of the nicer specimens that I collected, although it's missing its stalk, which I probably removed. Again, in the preparation of these things, it's fairly easy with an air abrasive unit. Occasionally you can find little bits and pieces that weren't collected in the 70s and the 80s. This last summer, Austin Heitzman found some cobbles in the stream that actually have some material that resembles this. Here's another slab that I prepped out in the 1970s. Those are now in at University of Oregon. You can prep out the stuff that is the original stereome. It's a lot of work. Unfortunately, most prepping these days is done simply by splitting the slabs, and you end up with kind of a upper and lower section of the animal, and there's a lot of information that's theoretically lost that can be put back together by putting these slabs back together, but these things are kind of like wood. They warp slightly. So one of the things that's, again, a future thing that somebody needs to do is to take a block that still has a bunch of this stuff in and take it to a facility that can do micro CAT scanning and actually get the animal three-dimensional and stuff. And why to do that is there's parts of the anatomy that we haven't seen. We need to study the 2 species. There's Isocrinus oregonensis and Nehalamensis to determine if they're really 2 species. We have to determine the genus, if Isocrinus is the actual genus or if it's another one. There was an attempt to rename it Remondocrinus, but the paper that did that did it in about half a paragraph. And it was by a Russian taxonomist who was paid by the taxon. You all know who this is. This is, okay, I was kind of like ready to not do anything with Mist this year. Austin contacted me through Bruce and wanted to go look at Mist and wanted to know where the crinoids were. And I thought, well, I don't know how many more times I'm going to be able to go in there. I have a lot of irons in the fire. I'm quote unquote retired and busier than ever. When I have the energy, I have energy ups and downs and stuff, and something called cyclothymia and stuff. And so um, going into Mist is something, plus I've gone in there a lot. It scares me. That bluff scares me. So he contacted me and he went out into the field and we were there FaceTiming and I showed him where to look. He collected this specimen and he's been collecting a lot of stuff and showing it to me, sharing with me a lot of his stuff. I'm really enjoying our collaboration, and he's just a very superb and wonderful person who I'm very, very happy to have met over this past year. So the conditions at Mist are actually quite crappy for collecting most of the time, unless you're collecting things people have left behind, or there's been a big flood that's washed all of the sediments away. But most of the time, the sediments covered. Now, I'm going to point out this little piece of tuft of grass up in the upper left corner. Oh yeah, this is usually what I walk away with, and actually in this pile, I walked away with some of it when I was there in October with Gary and Austin. They ended up carrying out a bunch for me, so, which was nice. It's great to have Sherpas. Here's the entire locality from across the stream. One of the things I want to point out over here, notice how the stratigraphy bends up. Here's the landslide chute, and the locality, the crinoids are mostly right around here, although we find other fossils, including fossil starfish up around here. There's a little lens of stuff here that's currently covered. Occasionally we find fossils out in the river. And I think Gary collected a slab somewhere out here. No, I don't want to copy that. Um, and also you can see a little line here, maybe. The stratigraphy bends up here. And you know, I thought that that was just a fold, and then oh yes, and here's conditions. This was right after the slide. This is my pack, which is about a meter tall, and the crinoids were oh, probably about 6 meters deep. So this was that slide in 1995, 96. So here's that tuft of grass that I pointed out. The crinoids are way underneath there. This was in 2011. It was there. Uh, yeah, that stick points to approximately where the crinoids are. There's that tuft of grass. There's the best crinoids are actually underneath this tree. Unfortunately, this tree sticks out on a promontory and is about ready to crash down someday. When it does, it'll make a huge mess. But once that washes away, it'll be a little bit safer to work. That's actually where the crinoids were the most dense, and that you could see them in cross section on the wall. I was there in 2011 with this character. This is Dr. Rich Moy at the California Academy of Sciences, who I met in the mid 1990s, and he is one of my co-authors on my Mist paper. He's also one of the world's leading echinoderm experts, and we have a few more papers that we're working on. This is Mihiro Matsumoto. In 2015, they snuck into the Pacific Northwest, and we kind of didn't want anyone, including NARG members and NPA members, looking over our shoulder. But we did want to attempt to dig down to the locality. So we went in there and dug about 2 meters down into uh, the into the talus and found the very first crinoids in situ from the Lagerstätten that had been seen since before the landslide in uh, in in 1995. For years and years, I've been really, really protective of this locality and, wondering how much crinoids are there to study and, and if any science could be done because it had been fairly heavily collected in the 1970s and 1980s. A lot of that by Portland commercial collector Paul Kirkland, whose material occasionally shows up and is being used in science. I have a picture of one of his specimens later on, but this was an amazing thing to see. One of the things I want to point out, this is the original stereome or the original calcite. The matrix, when it comes out, is very nice and dark when it's wet, but then as it gets dry and as it gets covered with talus and stuff, these crinoids are almost impossible to see unless you know exactly where to look. So sometimes you're lucky and there won't be any talus at all. And Tatsuo is standing right on top of a bench where we had collected a few years before. This is Tatsuo here. This is Moi Kato, who is another echinoderm researcher. Her specialty is crinoids and cold seeps. Tatsuo has a really, really cool shirt. So,
Mm-hmm.
and this was last fall. This is Austin and Gary. This is how deep they had to dig through the talus. And it was well worth their effort. They brought out one of the most magnificent slabs of crinoid materials that I've ever seen. Notice the tuft of grass up here. So you can see how far that they've had to dig. This is what they brought out soon after, and there is so much data in this piece, and it's like each of these crinoids shows various modes of attempted survival or mortality. Some of them preserve life positions. And it would be really, really nice to collect visual data of all the crinoids and, somehow look at that using experiments with modern crinoids. Uh, these guys dug this big pit and extracted the slab, which is all probably filled full of talus by now. But Austin has continued to go into the locality, and even when the water barely allows access, then it's been finding all sorts of interesting things this winter. One day he looked into the pit and he described it as like the trenches in France in World War One, and there was a dead salmon in the hole that they had dug. So these are some other people who have been important in Mist. This is Bruce Welton. He's a ichthyologist, and he went to work for industry for several years. And now that he's retired, he actually started working on a lot of Paul Kirkland's material, which is now down at a museum in Arizona. And he's working on trying to describe all the fishes that come out of here based on things like trace or the ear bones and teeth and scales. And using the fish, he's able to give us a constrained depth that's fairly accurate. Fish are fairly conservative over time in terms of what depths that they live in. And somewhere around 1,200 meters was the depth of deposition. These are some other people. This is Tatsuo and Moi. This is Elizabeth Nesbitt and her husband. She was the former curator of invertebrate paleo at the Burke. This is David Taylor. He was the first person who brought me to Mist in 1970. He and Bruce Welton have been kind of like long-term paleontological mentors. This is again another wonderful person. This is Carol Hickman down at the Museum of Paleontology at UC Berkeley. This is just pictures of posted of stuff out of her just outside her office at the museum. Here we are, happy crinoid researchers having just discovered the Lagerstätten. Here are some of Mahiro's experiments. These were modern crinoids that she collected all tangled up in the nets at in the harbor in Nagoya. And these things live forever and are hard to kill. So they'd been in the nets and exposed to the air for days. And she'd stick them in an aquarium where they would survive. And then she would subject them to varying quantities of volcanic ash to see what would happen. And sometimes they dig themselves out. Other times they would autotomize part of themselves and then dig themselves out. And sometimes they'd give up. Sometimes what they would do is that they would drop their arms off. And the arms have muscles on one side of these are some arms. Arms have muscles on one side and they have ligaments on the other side. Actually, the ligaments are on the outside of these curls and the muscles are on the inside. And I forget, may have that reversed, but what happens is that these autotomized arms form rings when one relaxes and the other kind of binds together. And the creepy thing is that these arms, they walk in circles for weeks, you know, as like crinoid zombies, whereas most of the animals still alive. This is one of several fossil examples of these things. So when you're collecting crinoids, if you see these as arms, something caused that animal some stress. It dropped its arms, and those arms walked around and for a while until they were covered up. Here's a LiDAR image of the locality. That the stratigraphy that I pointed out kind of goes like this, kind of a broad U shape here. Look in the lower corner of this screen, you can see the north-south orientation of this. I think what we have there, that what you're seeing here are the kind of broadly expressed shoulders of a U-shaped structure that runs kind of almost parallel to the outcrop. And that U-shaped structure, I'll trace with my pointer here approximately where it goes. It goes into the bluff. And I think that that's where the crinoids go. And that what we have is like a U-shaped scour channel. And it explains why we find things like leaves and stuff only in that spot. Otherwise, we'd find fossil leaves all over this thing. So we had a little channel with a current, which the crinoids liked. It might have exposed some hard grounds. And they thought, hey, this looks like a good place to sit. And then more volcanic ash came and dropped more stuff until it built up this layer. There's also a fault that runs through here too. You know, the geology of this area needs to be examined on a very, very high scale. Of course, these are projects for people to do generations from now. So here's some of that stratigraphy leading up to the south. It's similar to the north, but I didn't have a good image of that. Here are some more people I was out in the field with. This is the great John Armentrout, who's one of the great paleontologists and stratigraphers of the Pacific Northwest. And here is Alan Neem, who taught at Oregon State University. He was showing us some mapping that he did of the Keasey Formation, and the Mist gas field is actually very, very deep, and we don't think that that's the source of any kind of methane that came from that. This is something that John and Alan found. This again is something that needs to be mapped very carefully and studied, and possibly involving cores right underneath where we collect the crinoids. These are little tube worm traces, or not tube worms. They're called helminthoides. They're a trace fossil, and this is in a very carbon-rich layer that they found underneath. It may be that these things were attracted to a layer of woody or plant debris, basically ground up flower plant debris that had been brought in by lahar action, and then colonized by these animals. And it may be that some of the methane that we're getting from is simply from the decomposition of these, or they may have simply farted. And when the methane hits the seawater, calcite forms. Things that intrigue me are things like this. This is actually underneath Gary and Austin's slab. This is just kind of like a debris field of crinoid material. In this is what I call packstone. One of the ways in which crinoids will form a hard ground is simply by falling apart and leaving parts of themselves that eventually forms a gravel that they can stand on. So we collected some of these in October. And underneath that big tree, you can actually see a layer of this stuff with crinoids above and below. This is one of the Paul Kirkland fossils. This was collected in the 1970s. It just came to publication last year. It somehow ended up in Canada. I'm not quite sure how, but I'm really happy that this specimen, which Paul told me about years ago, finally got described. It's one of the prettiest fossil fishes I've ever seen coming out of the Pacific Northwest, and it's not impossible that there are more fossil fish out there of several different species. The fossil leaves, there's several species of leaves that have been found there. Nobody had studied these. And what could be done in terms of assigning this to Eocene or Oligocene is maybe do the paleobotany, including in these sediments, which preserve a lot of stuff, looking for things such as pollen. There's other weird stuff there. These were described as worm tubes or pogonopherans. I don't think they're that. I think that they're dentaliums, but and then I wonder, what is this thing here? Those I don't think are echinoid plate parts. So this is a specimen that's at UC Berkeley. A rock like this has all these little bumps and things. All of those might have interesting information. I'm going to show from here on kind of smaller and smaller stuff that intrigues me because it's easy to collect, doesn't require a whole lot of material, and I probably have more than I need. Of course, selenia, those are, you can find. Here's something, we don't know exactly where this layer is. We find these pieces, they've come falling down from above. We need a drone pilot on a day in which these things are coming down. I've looked with cameras and telescopes and stuff. We still don't know. Occasionally you'll find large blocks of these. This piece was about maybe 8 inches across. And
Austin and I did find a layer that has those urchins in them.
right, there is
You know where it is.
yeah, there is probably over on south of the crinoids. Of course, then we have things like here's a tiny little a clam next to a starfish and next to a squat lobster. We just heard from Carrie Schweitzer, who looked at the specimen, and she says, keep an eye out for more material. This is another specimen that Austin collected. There's fossil crabs. Some of these are not coming from the crinoids. They may be washed down. Carol Hickman had a comment about the fossil crabs and the crab collecting. Keep an eye out for things that are actually attached to the carapaces of these things. You know, those could be things like deep water barnacles and other kinds of epibionts and stuff. I know getting a very pretty crab specimen is nice, but you know, in the process of doing this, you may be losing some additional discoveries. Here is something that I found in a collection that was given to me by members of the Northwest Paleo Association. And we thought, what the heck is this? And then I saw an article describing carnivorous sponges, such as this one. And this is the first fossil record of a carnivorous sponge. And so this is another thing to keep an eye out for. In terms of sea stars and brittle stars, I know that lately there's been a lot of those coming out. The brittle stars, Gordon Hendler down at the LA County Museum talked about describing these someday. He has some beautiful material, but none approach the size of what some of the ones that are being found. There's sea stars. These were all done for Daniel Blake's doctoral thesis back in the 1970s. He said at one point to me that he'd like to redo those, but I don't think he's going to have enough time. He's pushing the years, and you know, so one of his students or grand students will probably reassign those, and hopefully they'll get specimens that are in good shape and not partially destroyed like this one that I did. This is what happens when your search image is crinoids and you don't realize what you're digging through. So I got rid of this arm, got rid of this arm, was working on this arm. I thought, what is this stuff? And I showed it to Dave, and Dave said, you idiot, that's a sea star. so sea stars are much rarer than the crinoids. So this was back in the OMSI days. This is something that bothered us for a bit. We didn't know what these weird triangly things were. Now, this is something not found at Mist. This is from a locality closer to Keasey, Oregon, that I have just GPS coordinates for. And Bruce Walton looked at those and said that they're pufferfish spicules. And so if you look at, you know, the external skeleton of a pufferfish, you see these weird triangly things that look like, you know, little tetrahedrons. So you'll find a lot of mollusks there. Fortunately, we can assign names to most of them. I've called this Solomaya. One of the things I wanted to point out this specimen is notice the soft-bodied preservation. These things have a kind of a chitinous exterior on them, and you can see some of that, you know. So looking for soft-bodied stuff is something to be aware of at such a locality as Mist. And you get messes of things. This is from that upper concretionary layer. What's intriguing about this is this stuff. We think this is part of an Eturia and a lot of Acilla, Nihilamensis. This is something that's relatively unstudied that when I go out there next summer, I'm hoping to collect from that layer, assuming I can get to it. It's usually about 8 feet above the head. Last time I looked, it was about a meter or two underneath the sediment. And in 2017, there were 2 big logs laying on top of it, and now there's blackberries. These are some pteropods, which are little. I found this in this big concretionary mass that's out in the middle of the of the stream.
Hmm.
These, I think, got described by Jim Geddert years ago. And then these are thin sections. This is stuff that hasn't been studied. What is that bright thing? Well, let's zoom in. That's a radiolarian. Here's something for somebody to look at someday. I'm not going to live long enough to get into all of these things. On the microscopic level, mist is a whole lot of fun. Even weathered matrix, you can find like a lot of foraminifera. This is what some of the crinoid material looks like. You can see the individual cells in the stereome. You can see articulations where they come together, and these are probably columnar parts, more crinoid parts in cross section. I don't know what this is. It might be some crushed micromollusks or something. Another view of that. There are paleontologists who get turned on by weird stuff. This looks like a echinoid spine in cross section. It might be that these are stereomes, so these might be echinoid parts. Little bits of spine. Forams. The forams of the Keasey Formation have been studied variously. In cross section, they're actually quite fun and quite crazy. These are sections that you have to basically grind a piece, polish it, glue it to a slide, and then grind the other end until it's about 1/20th of a millimeter thick, and then look at it underneath the microscope. These were all done for our 2005 paper. So, and the tiny stuff can be significant. This is another locality that I've been working on since 1990s. This is a teeny tiny echinoid that we're now calling Neofibulariella, and they're really tiny, as you can see. Sometimes the tiny stuff is interesting. Jim Geddert found this very, very easy to get to locality. We had to have a collecting permit. We collected about 40 of these things. Rich is now in the process of doing the final bit of work to create a paper. And so even for a little tiny echinoid, it can take a long time before stuff gets published or even identified. One of the things that we're discovering from this, there's a European and African genus called Fibulariella. He's recently divided the ones in the West Coast to a new genus. I'm not supposed to mention that. But in order to describe this, he has to go through the entire genus of modern and fossil specimens just to put things into context. One of the echinoids that Gary brought in tonight is interesting to me because I'm hoping it's something like this. This is a poster that was done based on research from some of the observations that Rich Moy and Chuck Messing made off of the Johnson Sea Link submersible. We found evidence they found these sea urchins. This one's called I forget. Anyway, there's like 3 or 4 different genera of sea urchins that their diet is basically chewing on crinoid columnals, and so the crinoids don't like that and they run away, and so this was a poster done, I forget the name of the artist, San Francisco artist who did some artwork for the Academy, and just showing this relationship. So anyway, the thing, just to conclude, Mist is a locality that has a tremendous amount of potential for future research and discovery. I'm winding down what I can do with it just because I'm getting too old. reach average male life expectancy in 5 years. And it'd be nice to have another lifetime to pursue all of this, not to mention a budget for it. But so I'm kind of in a mind passing all of this stuff on to the future for people to work on. Mostly people will go there and enjoy collecting it, but it's important to document where you find things at the locality, photograph stuff in situ if you can, or note that it's float. And then when you're done with it, don't post it up on eBay or anything. It's a good idea not to monetize fossils at from such a precious locality. Otherwise, um, um, there could be gold rushes and suddenly a lot of competition. Instead, find a museum that's willing to take this material. They may be it may be getting full up on crinoids, but there are always other museums who are interested in the material. It's just it makes it accessible to researchers. I'm glad the University of Oregon is willing to take material. The Burke Museum is willing, as well as the Museum of Paleontology in Berkeley, California Academy of Sciences, as well as the LA County Museum, which has a tremendous amount of missed material. Paul Kirkland used to work there as a preparator and sent some very, very lovely stuff down there. So anyway, questions?
Thank you very much, Keasey. Last thoughts on how we as a, club should treat this Lagerstätten. You mentioned a few things right there at the end. Don't sell them. Uh, don't make a big deal of it. Try to keep it a little bit undercover. But what else? Because this is a very special spot, and
It,
we've done some stuff in the past with it, but we're going to continue to do more.
right.
What do you think we should do as a club to preserve and best utilize this Lagerstätten?
Are you guys familiar with the book Fossils in Oregon by Margaret Steer? This was published by the Department of Geology and Mineral Industries. It was basically a collection of articles from the Orbin, which was their monthly publication. Margaret Steer, she was actually my very first paleontological mentor. I knew her from like age 8. She wrote about this locality, the rarity of these crinoids, which is again reiterated by Warren Vokes, and that it deserves some respect. And in the modern terms, in terms of our access there, it's on state property, it being a navigable waterway that is by 1859 standards, when Oregon became a state. I think it's 1859. Um, the road that you drive in to park there is still, I think, a county road. The land that you cross, though, to get to the river is private property, but it's owned by some trust, and it will probably be logged at some point. Um, you know, I would try to make a habit of just going in there very discreetly and but not frequently, otherwise, the neighbor along the road there might object. And you know I don't know what the status of that road is, if the county has voided it, and if it's a private road. I know that there are more properties and stuff, so you know some discretion. I know that in the past, the property owner across the stream, Mr. Epling, has allowed access, but then you have to cross the stream, which has some danger to it and stuff. And most importantly, when you go there, wear a hard hat. That locality is always sending stuff down. avoid going there after heavy rains and snows and stuff, even if the river is low and tempting, because that's when stuff is likely to fall, and stuff. But generally, it's a difficult locality to go and collect in. I know that was a lot of work that Gary and Austin did to get those big slabs out. I did that early on when it was easy. There was actually a bench where the stuff stuck out like a sore thumb. And but even back then, we were all kind of careful not to be, make a whole lot of fuss getting in there and out there. So um, so my recommendation is uh, treat it lightly, collect some nice stuff there, but at a certain point, we may decide that we have enough crinoids for this generation and that we want to save it and access for the future generation, and explore other localities which are accessible. So,
Mm-hmm. Mm-hmm.
Anything else from the people online?
Yeah, I've got a question. So I know you said that they, shed their arms. Is there any sign of regeneration on these specific
oh yeah.
specimens or predation in general?
Oh, I don't know about predation. We're still looking to see if the echinoids, are eating any of these things. Certainly, fish and stuff would come along, and you do find a lot of specimens with regenerated arms, that are sometimes much smaller in profile and stuff. Sometimes you'll find specimens where the arms are all down autotomized, almost to the stem, and I have one specimen that has like 3 or 4 uh, plates on the stem and about maybe 3 or 4 plates above the cup. And it automized a lot of stuff, and whether or not that was response to being eaten or survival, I'm not sure. The animals, if a little bit of the cup is missing or is still there, the entire animal can regenerate. So, and again, that's another thing that looking at hundreds of specimens that have been collected over time, that could give us an idea of, how much of this was simply trying to survive ash falls or trying to survive fish, because you wouldn't see regenerated arms in a specimen that was killed by the ash falls. That's a good question.
Thank you very much, Keasey. We appreciate your insight and your history. It's really cool. And when you get a date that you're coming out, please inform us so we can come out with you.
I would like that. It'll probably be late spring, early summer. I have to get myself in shape. I've been kind of too sedentary this winter, so
Okay, can you
yes,
take and make us a host again?
yes, I will do that. Let me.
Yeah, turn off this.
Okay,
Sure. Yeah.
let me get rid of this and restore Zoom.